So I think I've always had this innate interest in how things work, and I still have on my bookshelf at home the book of science experiments that my dad bought me for my 9th birthday, which I seem to remember involved a lot of playing around with vinegar and bicarb and fairy liquid. My parents and teachers were fantastically supportive all the way through, which I was really lucky with that. At school I thought I probably wanted to be a medic, but over time I came to realise more it was really the sort of biochemical underpinnings of health and disease that were what really interested me and really inspired me, so I decided to pursue biochemistry at university. Over the summer holidays I was really lucky to be able to do some placements in the laboratories of inspiring women scientists, and learnt the basics of how to hold a pipette and how to chill a bottle in an ice machine, and those really strengthened my resolve to do lab-based research after my degree. So I stayed at Oxford to do my doctorate in structural biology with a particular interest in molecular immunology. Because I had this fantastic immunology tutor during my undergrad degree, and during my doctorate I stubbornly persisted with an immunology project which was going a lot less well than another project I had going in parallel. I always knew that the greatest area of interest for me was research with the potential for clinical benefit to patients. So I started looking for industry-based jobs towards the end of my doctorate, and a job at Adaptimmune came up at just the right time. So here I am. In a few weeks' time I'll actually have been here for a decade, which definitely makes me one of the Adaptimmune old-timers. With apologies to everyone else. When I first started here in October 2012, I was a scientist with a focus on molecular biology and protein biochemistry, and I'd been hired as part of an expansion to around 15 people, so we were still pretty small, and I really enjoyed learning the ropes of a growing company on a small team. At that time, all of Adaptimmune were in building 57, which is one of the older buildings on Milton Park, so the move of a few groups recently back over to building 59 really felt like quite a homecoming for me, so that was nice. One thing that's changed a lot in that time is the adoptive cell therapy field, in particular the number of companies working on TCR T-cell therapies. When I went to my first conference shortly after I joined the company, CARs were definitely the thing, and Adaptimmune was the only company working on TCR-based therapies, and that's certainly changed over the course of time since I've been here, and it's been a real privilege to be kind of part of a fast-moving field with such a lot of interest in it over that time. I remember when we signed the GSK deal, Helen brought round some champagne on around the single office that we were all in then, on a stores trolley, which was definitely a great celebration. So I'm now a principal scientist within Marc Lenoir's research tools group. We're a tight-knit group. It's a real pleasure to work with such a great group of people. But we work in a really highly collaborative way with colleagues from across research development to understand what tools they need to further their research and to leverage a range of technologies to provide those tools. And that could be anything from using gene editing to create new cell lines, or it might be leveraging protein expression expertise to supply key reagents for some new experiments. My particular role within that sort of that context is to be a bit of a wise woman of molecular biology, to help colleagues kind of understand and access our plasmid reagents, and to design and produce new ones when they're needed by people's research. In the course of that, I've sort of developed expertise, which has meant I've contributed to sort of regulatory paperwork as well, which is kind of given me a wholly different perspective on that side as well. In parallel with the molecular biology work, I've been developing an interest over the last few years in understanding more about how we might make modifications to our TCRs which might universally boost their potency. And one way of doing that is to find variants or modifications which universally boost the surface expression level of TCRs at the cell surface. But to do that, we really need to understand better surface expression levels, so that we can see which modifications are making a positive difference. And that overall, that forms the basis of the TCR optimization initiative, which I'm leading and working on with brilliant people from across a lot of different groups, which is great. It's really great to have an opportunity to collaborate with lots of different people from across different groups and get different perspectives on what other people are working on as well. So I'd only been at Adaptimmune about a year when I put in a very speculative application to present a talk at PEGS Boston, which is one of the larger high-profile conferences in the field, particularly at a time when there were fewer specialist conferences on adoptive cell therapy. And to my surprise, followed by delight and absolute terror, my application was accepted, and with a lot of help and a lot of PowerPoint animations, I put together a talk for the conference. And I think I was the night before my slot, I was up till about 3 a.m. Practicing my spiel, powered by pretzels and Diet Coke from the 7-Eleven across the road. Yeah, I think on the whole I'm pretty good at holding my nerve, but I've never been more nervous than when the conference assistant was pinning my microphone on. But thankfully the talk went quite well. And that evening, Joe Brewer and some others took me out for a meal to celebrate. So that was certainly a trip to remember. Well, at the moment I have an 11-month-old baby daughter, so pretty much outside of work what I do involves stacking cups on repeat, moving my phone beyond crawling range, and reading The Very Hungry Caterpillar again and again. But outside of that, I've loved singing with others since I've been at university, and that's everything from Renaissance and Baroque polyphony to through to modern a cappella, and everything in between. So I'm really excited to have rejoined my chamber choir, which is based in Oxford. I used to do some coxing of rowing boats, but where I live now in Farringdon is quite far from any rowable stretches of river, so I've had to give doing that up on a regular basis. But having done the Couch 5K program here at work a few years ago, I'm excited to get back into a little bit of running. But fortunately, even having a baby couldn't interfere with my love of food and cooking, so I'm always enjoying trying out new recipes, and over the summer I even made some ice cream for the first time. During 2019, I took advantage of an opportunity to do a secondment placement in the Allergenic team with Garth Hamilton, which was fantastic, and I learnt so much doing that. It was a really brilliant thing to do. One of the projects that I was working on during that time was to produce a knockout of a particular gene which would make the pluripotent stem cells, which we work on in the Allergenic project, more amenable to further modification with our engineered TCRs. That required a lot of tissue culture and some very painstaking clone screening. So my microscope eye and my pipette arm certainly both got a very good workout. Towards the end of that, I was looking to isolate clones in particular that had both copies of the particular gene knocked out, and I remember quite clearly when I was looking through the sequencing results, I could see several clones which looked really good but had only one copy of the gene knocked out. So I was trying to keep calm and go through and just keep going in the hope that there would be one with both copies knocked out. And I remember when I saw that one of the clones had both copies knocked out, I probably startled quite a few people in the office by yelling "yes!" And then once I calmed down, I went back and checked and rechecked to make sure it was true. First of all, I would say don't be afraid to ask questions. Personally, I believe there's pretty much no such thing as a stupid question, and I've learned so much from hearing how other scientists ask questions about people's work and thinking about how to answer questions that other people might pose about my work. Asking questions can open up whole new sort of thought processes or lines of inquiry, whether they're sort of why we do things a certain way or just to make sure you've understood something. It's always worth asking that question. Secondly, collaborate as much and as often as you can. I think it's really easy, particularly in a larger organisation, to kind of compartmentalise or feel that you really need to focus on your own work. And obviously it's important to get done what you need to get done, but where you can, just really try and widen your perspective and try and see what else is going on and trying to develop your understanding of how your work fits in with what other people are doing. And if you get an opportunity to work within another group or kind of reach out across boundaries, then I would always recommend do that. And my final bit of advice is to find something that you're really interested in and work on it. Work on understanding it as hard as you can and enjoy doing it. And if you're really lucky and get to work with some brilliant people who motivate you and inspire you and make you laugh, then that makes everything so much easier.